Welcome, everyone. Thank you for joining us today. I'm Tracy Smith, the Program Director of Education and Outreach for the Center for Integrated Cellular Analysis here in New York City. In this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. And today we will discuss GoT-Splice, a multi-omic approach that uses long-read sequencing for single-cell transcriptomics and proteogenomics. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. Our guest today is Mariela Cortés-López, a postdoc researcher in the Landau Lab at Weill Cornell and the New York Genome Center. She earned her PhD in biology from the Johannes Gutenberg University of Mainz, Germany, where she studied the influence of mutations in splicing of cancer-relevant genes. Mariela's current research focuses on dissecting the cell type-specific mechanisms of RNA regulation by integrating long-read sequencing technologies with single-cell multi-omics assays. With that, welcome, Mariela. I'll hand it over to you.
Thank you, Tracy. And hello, everyone. Thank you to the Center for the invitation. So today, let me share my screen. Okay, I hope you can see it now. Okay, so today I'm going to talk to you a bit about this recent publication that we put together in the Landau Lab, where we are trying to identify how splicing aberrations can be studied at the single-cell level. And this is important because most recently, it has been described in many publications and through many tissues that our cells present non-identical genomes, which means that the cells that we are born with, they acquire mutations when we age, and then most of our tissues look like mosaics. So this accumulation of mutations that happens with age results in different consequences. So particularly in the hematopoietic stem cells, so in the blood system, sometimes some of these hematopoietic stem cells can acquire driver mutations, which along the years, then they can result in the development of hematopoietic cancers. So, for example, leukemias and myelodysplastic syndromes. And within this hematopoiesis process, splicing regulation is also something that is very critical. So it has been described in several studies how upon the different steps of regulation of hematopoiesis, which is like the process that gives rise to the cells in the blood, some changes in splicing will have consequences leading to the production of certain proteins upon a specific time point. And therefore, the regulation of splicing in hematopoietic systems is very critical and often is affected. And splicing alterations are very abundant in blood cancers. Here, I'm showing you a recent review where you can see that in some particular blood cancers, some particular variations of myelodysplastic syndromes, we see a high frequency of splicing factor mutations, sometimes even going above 70% of the patients. And particularly, these mutations tend to affect mostly SF3B1, which is a core splicing factor that recognizes the branch point and then helps to identify the 3' splice site in an exon. But we also see an increase of mutations in other critical splicing factors such as SRSF2 or U2AF1. We became interested in studying SF3B1, mostly because SF3B1, when it's altered, so normally SF3B1 will recognize this branch point, work together with the U2AF2 and U2AF1 subunits of the spliceosome, and proceed to have a correct splicing. However, when you have a mutation in SF3B1, what happens is that SF3B1 will recognize then an aberrant branch point, and this will lead to the selection of a cryptic 3' splice site, which results in a transcript that contains a little bit of an intron, and therefore this tends to be often to disrupt the frames of reading when it's producing a protein, which will result in the production of aberrant proteins, or sometimes sending these aberrant transcripts to nonsense-mediated decay, which also decreases the production of the normal protein. Therefore, understanding there's a need of understanding how, particularly in the different cell types across this differentiation and hematopoiesis, how the aberration in splicing factors can be affected, and what are the consequences. So we want to know, we want to study the relationship between splicing aberrations and the observed cellular phenotypes. But however, this has been very challenging across the years. First, because there are no very good mice models that can also replicate what we observe in human cells. And also, even when there are some existing cellular culture models, it can be challenging first to produce a proper cellular culture model that will recreate what happens in the human cells. And on top of that, there are other more technological limitations to study splicing factor mutations directly in human samples. Starting because most of the studies so far have been done in bulk samples, and bulk samples always have the problem that you can have a high cell heterogeneity, and then you cannot distinguish which are the cells that have the mutation and the cells that are wild type. And moreover, when you want to go to look at the individual cells in the hematopoiesis process, every cell has a complex cellular phenotype. So when you study everything together in bulk, sometimes you can miss the particularities of certain cell types. And even though there has been an improvement in using single cell approaches to study these complex cellular phenotypes, the fact that most of the approaches relied in short reads also implies that when we want to study splicing, we will have incomplete reconstructions of the transcriptome. So in the lab, we have been focused very much on study cancel evolution through a multi-omics perspective. This means looking at single cells. We want to see at different levels, how can we study different things in a single cell. So mostly, many groups have focused mostly in mRNA and studying individual variations between cells. Here, we also want to integrate other aspects of this variation to study, to give a more complete view of the cells. And in this case, we also added protein expression to the study of particular cells in the hematopoietic system. This resulted in a new technique that we call GoT-Splice. And GoT-Splice will retrieve genotype of the transcriptomes and splicing at the single cell level. So GoT-Splice is based on the previously developed assay in the lab known as GOT, or genotype of the transcriptomes, which basically relies on the 10x genomics platform in order to obtain to genotype particular cell types. So we divide the full-length cDNA library into 3 parts. One that will be the short-read library together with the CICA, which is a technique that was developed in the ICA, in the sex ICA by the Cellular lab. in the New York Genome Center. We also integrate this previously developed GoT technique where we target a specific mutation in every single cell. And we included here the long read sequencing in order to have an accurate reconstruction of the transcriptomes. So at the end, after having these three levels of information, we will get a proper transcriptome from every single cell, which protein marker, so the markers of the surface that will let us know specifically which are the particular cell types that we have in our samples. Using the long read, we will have a proper reconstruction of alternative splicing processes because we will obtain the full length transcripts. And using GoT, targeting the specific mutation of interest, we will be able to distinguish the mutated cells versus the wild type cells. So we started the study with a particular cohort that contains mutation on the SF3B1 gene. And this cohort is has the disease myelodysplastic syndrome, which is very frequent, which frequently contains this particular mutation. And our initial cohort was of 3 patients. So from these patients, here I'm showing you some of the results that we got with GoT-Splice. We recover most of the cell types that are expected to be seen in the hematopoietic system of these samples. So we also observe that there is an increase, something that looks like an accumulation of erythroprogenital cells, particularly in these MDS samples. And this can be observed also when we look at the genotyping of the cells, that we see that most of the mutated cells are also matching with the accumulated erythroprogenitors, giving us already an idea of what can be the disruption that having this mutation in SF3B1 causes to these particular patients with MDS. Moreover, using the CICA technique, we were also able to identify the cellular markers present in the surface of the cell types, and also distinguish some cellular markers that might be more abundant in the mutant cells compared to the wild type cells. So what we described was that we observed that CD71, which stands to be a marker for erythroprogenitors, is a little bit more abundant in erythroprogenitors when we compare the mutant cells versus the wild type cells. And we also observed this increase on the expression of particular cellular markers at the different stages of the trajectory and differentiation in hematopoiesis. So we see that late cells that would be the most developed cells, then they would have an increase on the expression of particular markers that define the erythroid lineage, in this case CD36 and CD71. And moreover, the mutant cell fraction increases also with this trajectory, meaning that there is an accumulation of these mutant cells with the later trajectory times. And these mutant erythroprogenitor cells tend to accumulate and resulting in changes that are mostly associated with cell cycle and translation. This was an analysis that we obtained from looking at the gene expression, where we can identify that mostly in the erythroprogenitor cells with that are mutated in SF3B1, we observe an increase on the expression of genes that are associated with translation and also cell cycle. We can, moreover, quantify these changes and across the different cell types we were able to observe how there was a constant increase in the TP53 pathway. But now this all has been done with the SHORI data, with the genotyping and the site-seq data. But we wanted to see how powerful can be now to integrate the long-read data in this particular analysis. So we wanted to ask the question whether GOT is able to reveal the mis-splicing phenotype described in these MDS samples. First of all, I would like to bring to your attention that in order to have a better capture of the long-read data, we apply some improvements to the normal, to the initial ONT long-read technique, because given that this will aim to capture the full-length transcripts, sometimes many times the incorrect capture of transcripts that have an incorrect structure can be more abundant and overcome the goals to capture the full transcripts. So a full transcript will look like this, in which we have the nanopore adapters at the end. In the middle, we would expect to see the full-length transcript, followed by the cell barcodes and other adapters that we need to obtain the information of single cells. However, just adding a little bit of biotin to capture the ONT adapter increases the correct structure capturing of the reads above 75%. And we see that most of the full-length transcripts that are captured now can be preserved only with this particular biotin change. This has been implemented before in other ONT single cell assays, and now it's a standard after this study and others. Now it became a standard for the nanopore protocol for single cells. Moreover, now having long-read sequencing will provide more information at the single cell level for the individual transcripts, because normally in the 10x genomics protocol, we will have only around 50 base pairs of the cDNA. However, when we use the long-read protocol, then we extend this window of capturing a transcript, and now we will be able to get high resolution of more than 1 exon or 2 exons, and be able to call these particular variations in splicing, in this case, that we are interested to see. This is, for instance, one example of how the capture becomes improved using the long-read sequencing. You can see here in the zoom-in how we were able to detect a particular cryptic triplet splice site in the long-read sequence only, compared to using only the short-read sequencing in yellow. So now, having this a better capture of full-length transcripts, we went to our data to see how the mutant cells could show an increased cryptic triplet splicing. So in this case, we compare the SF3B1 mutated cells versus the SF3B1 wild-type cells. And as you can see here, it's very evident how there is already a skew of all the transcripts that show a differential percentage splice in, or a differential usage of particular splice site, in this case being cryptic 3' splice sites, only present in the mutant cells and not in the wild-type cells. And this can be exemplified with, for instance, with this particular gene, the metal 17, where we can see how only the mutant cells we have the sudden increase of the usage of a cryptic splice sites upstream of the normal 3' splice site. Moreover, having the cell type information, now we were also able to follow the changes that happen across the differentiation trajectory in the hematopoietic system. So here, for instance, we are able to observe in early stages the different values of splicing changes across the continuum of erythropoiesis. And we can see how some particular genes tend to increase the usage of a particular splicing site with the increase on differentiation. Here I'm just highlighting a couple of genes that had been reported already on SF3B1 mutant cells, SF3B1 bulk studies, but also some new candidates that I'm gonna tell you a little bit about one particular candidate that we reported in this paper. Moreover, now we assess how comparing mutant versus wild type, we now have the resolution to identify these particular changes. But we can also now identify changes at the level of cell types. And this is what I'm showing you here, how looking at particular cell types, now we can group this differential splicing events and how they happen together in a particular cellular context. So for instance, here we could identify a particular cluster of genes that are only differentially spliced and strongly differentially spliced in erythroprogenitors. And as you can see here, many of these genes tend to have or have been reported to have a function specifically associated with erythroid differentiation, as well as other processes that will be critical to explain the phenotypes observed in MDS. So we also went and because our original cohort was only consisting of 3 samples, we wanted to validate what we observe in this cohort, given that sometimes there can be a sample variation. So we included also a MDS validation cohort, which also comprises other 3 patients that now will have some certain treatment for MDS. And we were able to observe that some of these events that are differentially spliced, they can also be recapitulated in this new validation cohort. Particularly interesting for us was one event in PAX, which I will come in a little bit to tell you more about what we found in PAX. But there is also other interesting events that also have been associated with the phenotypes of SF3B1. But moreover, in the study, we were also interested in to look into other condition that is known as clonal hematopoiesis, which also has great abundance of SF3B1 mutations and other splicing factor mutations. So this is normally when a cell acquires mutations and certain mutations become more prevalent, but before some disease develops. So this is observed sometimes in certain individuals when they are aging. So they tend to accumulate more mutations in particular genes, but they haven't yet developed a phenotype that is associated to a disease. In some cases, this can be considered as a pre-malignant state, and therefore it results very important to also understand whether the prevalence of this, in this case, SF3B1 mutations, whether they also have a conserved mechanism that can be explained then the development and the transition to a myeloid malignancy. Because as we can see here, some of these mutations, particularly splicing factor mutations, tend to have a higher risk in these patients or in these individuals to then develop an hematological malignancy. So we applied GoT-Splice to SF3B1 mutations, so a cohort of SF3B1 with SF3B1 mutations. In this case, two different individuals with two different mutations in SF3B1. In this case, we also recover the expected cell types that are observed in a hematological system. So we observe many HPCs, which is common to see in the CH. But we also detected that many of the mutant cells tend to be accumulated in the erythroprogenitors in these cells. It's a little bit less dramatic than in the MDS case, but you can already observe it in this pre-malignant condition. Moreover, when we now look at the trajectory of the differentiation in the CH samples, what we also observe is that similarly to what we see in the MDS cohort, most of the cells, the mutated cells tend to accumulate in late differentiation trajectories, mostly going towards myeloid and erythroprogenitors. Also showing already a differential phenotype that looks like MDS. And when we look at particular changes in splicing we compare it with MDS, we can also see some overlap of some of cryptic splicing, 3' splicing junctions between this pre-malignant condition and the MDS cohorts. Particularly some of the genes like metal-5 or URAT, which had already been described as being misregulated in SF3B1 mutants. And moreover, now I'm coming back to what I was telling you before, that we also see some very interesting new events that have not been reported before. And one of them is particularly this event in BAX. So BAX is a apoptotic protein, which normally it has different isoforms, being BAX alpha the most abundant. And when it is normally expressed, it will promote apoptosis. However, when a different isoform, in this case, is called BAX omega, when BAX omega is expressed, it tends to lead to the opposite role, which is very common, a very common mechanism that these apoptotic genes tend to have. In this case, BAX omega will lack a particular terminal domain of the normal BAX protein, and therefore will prevent the apoptosis of the cells. And what we observed in all of different cohorts was that in MDS, we can see a constant missplicing of BAX across the differentiation trajectory, which is also recapitulated in our MDS validation cohort. And slightly, but also increasingly, we observe that in our CH cohort, we also see this misregulation, which can explain potentially why the erythroprogenitor cells, the mutant erythroprogenitor cells tend to mostly accumulate, but they are not completely eliminated. They don't go to apoptosis as first route. And with help of some of our collaborators, we actually tested this function, this anti-apoptotic function that Bax omega could have. So in this case, our collaborators help us to test how Bax, the different isoforms of Bax can indeed trigger apoptosis. However, with Bax omega, we see that the apoptosis is never triggered at the levels of other particular isoforms of Bax. So more studies will be required to see how generally the role of Bax in together with other of the misregulated events are affecting the MDS phenotype. But we were able to see this at the single cell level and already an increase with the continuum of the erythropoiesis. And last but not least, I want to also show you a little bit what we included in the paper, how GoT-Splice can not only be exclusive to study SF3B1 or to study splicing factor mutations, but also how we are bringing GoT-Splice to other splicing factors and also other regulators that tend to be highly mutated in these myeloid malignancies. So here I'm showing you the example of U2AF1, which is also a critical core splicing factor. And we apply GoT-Splice in this particular AML sample, where we identify a high number of mutant cells that we compare to wild-type cells. And we were able to recapitulate the consistent change that has been reported for U2AF1, which is an increase in exon skipping. In the case of now going away from splicing factor mutations, we also look at DNMT3A, which is more epigenetic regulator, transcription factor, but which also sometimes these transcription factors tend to crosstalk with the splicing regulation. And in this case, we observe that also particular events can be detected when we compare wild-type DNMT3A genotype cells compared to mutant DNMT3A genotype cells, and using the long-read sequencing. So here as well, we can see that this particular isoform of SRSF3 gets more expressed only on the wild-type cells, but not in the mutant cells, which is also something that we are working on a little bit more on exploring other splicing factor mutations and transcription regulation affecting splicing. So in conclusion, I want you to remember that GoT-Splice can be very helpful to profile single cell genotype, expression, surface markers, but also, and more particularly, splicing. And that now we apply GoT-Splice to SF3B1 mutations, and we show that there is a particular cell-specific missplicing observed by SF3B1. And this impact of SF3B1 mutations can manifest before the disease onset and seems to be perhaps a conserved mechanisms that can already be observed in the pre-malignant state of CH. And with that, I would like to acknowledge my team and Dan also for the opportunity, as well as all the members of the Landa Lab and our great collaborators. And if you want to check the story here, I just put the QR code. I can link the paper in the chat, and I'm happy to take any questions.
Incredible talk. Thank you so much, Mariela, for sharing your work with us. And let's move on to Q&A. If anyone would like to ask a question, you can raise your hand or type your question into the chat box. But one question I wanted to ask was about your capture rate of cell type diversity in samples. How has it with GoT-Splice, is that similar to other techniques, or?
In fact, in the paper we included some metrics, and we see that there's a very strong correlation, more than 80% from the long read versus the short read. It always depends on the sample. Sometimes there are samples where with lower quality than the amount of long read versus short read UMIs that you can capture gets lower. But usually, if we have a very clean sample with the biotin approach, then we get a better recovery.
Okay. And do you use both the long read and short read data in generating your cell clusters?
Mostly we use the short read data. We are exploring a little bit and some derivations of this work, like using the long read data, but so far I think the problem is that also there are not many like very good resources where you can really differentiate or have a knowledge of certain splicing isoforms that could be more cell type specific, because still the long read data sets are quite low. But hopefully we could get there, improving also with the isoform information, the cell type annotation.
Right, thank you. Well, thank you so much for your talk. It's been very exciting. This seminar has been recorded and will be available soon on YouTube and on our website, multimodalintegration.org. Oh, we have one more question that just popped in here. So let's ask that one before we sign off here. So the question is, how sensitive is this method to detect genotype at a single cell level?
So with the genotype, it varies between the samples. With some samples, we get above 80% of the cell's genotype. It always also depends on the target that we are aiming to capture. So in this case, SF3B1, because we use the short read to do the genotyping. SF3B1, the mutation is very close to the end of the transcript, so we get a high efficiency of the genotyping. In other cases, it can be a little bit more tricky, and sometimes also we have tried to genotype using only the long read. The throughput is lower, but it can be increased sometimes around 10% or 20% more. So when we want to compare wild type versus mutant, we mostly rely on the cells that we are confident that we can genotype, excluding the ones that we don't know. But we are also implementing different methods to maybe infer the genotyping based on the transcriptome, or using other techniques like the recently developed GoT, which relies on the chromatin to improve the genotyping of targets that are not highly expressed. So those are sort of the limitations, the expression and the location of the mutation that influence the genotyping.
Well, thank you for answering that question. And thank you again, everyone, for joining. We hope to see you at our next seminar. Have a lovely day.
Thank you.